Hello again, biology. Welcome to part 3 of your notes on cell replication. In this section, we are going to be talking specifically about meiosis, which is also called sexual reproduction. Okay? Last time we learned about mitosis and like all that stuff. Binary fission, mitosis, cell replication to get 2 exact cells. Okay, that was mitosis. Now we're going to be learning about meiosis and how to compare it with mitosis, seeing how they're different. Okay, so earlier at the very beginning of the slideshow, it talked about learning goals. These are the ones we're going to be covering in this section, which is comparing and contrasting the processes of asexual and sexual reproduction, as well as the advantages and disadvantages of each. And then define, identify, and distinguish the differences between mitosis and meiosis. Okay, so like I said, meiosis is what we're focusing on here, and it is called sexual reproduction. If we have asexual reproduction and sexual reproduction, meiosis being sexual, that would lead you to believe that mitosis is asexual reproduction. Okay, so I'm going to go over quickly the vocabulary of those. Asexual refers to being able to replicate without going through a process of sharing DNA or like being fertilized. So that makes sense because your skin cells, they don't need to be fertilized or anything in order to make another one of itself. It just goes through the cell cycle. Whereas meiosis is a sexual reproduction, and it requires fertilization by another source of DNA in order to begin replication. Okay. Something that helps me remember meiosis is the word is what it starts with, which is M-E. Some people pronounce this meiosis. I pronounce it meiosis. But regardless, meiosis has to deal with either myself or me. So this is how we as complex beings, like humans and animals, come to be, is because of meiosis or meiosis. Okay. So let's take a look at what that entitles here. So first I'm going to review what mitosis is all about before we get to the new stuff. So mitosis, what was it? Mitosis it's asexual reproduction. It's where we had an exact cell being produced. That's the purpose of it, is to make an exact copy of the cell that you started with, as well as growth and repair. As I said, it's referred to as asexual reproduction. What's the ultimate result? That was the cell that is exactly like the other one. Okay. Here's your important vocab. It's coming back. Only you'll notice that we have new things that are underlined. So the stuff that's underlined and is in blue is what we're going through with this half of our notes. So we're going to be focusing on things like meiosis, sexual reproduction, crossing over, haploid, diploid, and the rest of those. Okay? So let's start out talking about somatic cells versus gametes. You may have not ever heard of these two before, but basically somatic cells are body cells. These are cells in your body that make up your body. Okay, body cells. Similar to skin cells or muscle cells or hair cells, nail cells, stuff like that, okay? Whereas gametes refer to like They're called sex cells. Okay, these are reserved only for making a new being instead of just a new cell. These help with making brand new people or animals from these, okay, through fertilization and stuff. Okay, our bodies have both types of cells, and both of them have their places, and they have their different things that they do. Okay, body cells, they're made out of tissues. Okay, they make up your tissues in your body. Things like I said, muscle, bone, skin, nerve, heart, brain, those are all somatic cells. Okay, we have 42 chromosomes or 23 pairs of these types of like DNA for these. Okay, and when that's what they have when they're finished replicating. They have 46 chromosomes or chromatids, and they're organized into 23 pairs. Okay? They are made via mitosis. Whereas gametes or sex cells, they're involved in sexual reproduction compared to being made out of making up your body tissues. These are specific cells. They're specialized. Examples of these are things like your sperm cells or egg cells. Okay, now we see that they're little individual things instead of big tissue things. Okay, when they're finished, like being made, we have a total of 23 chromosomes in them. Okay, they are produced via meiosis or meiosis. Okay, and then like I talked about, we needed fertilization to happen in order for us to have to have sexual reproduction. This is when a sperm and an egg come together to form a fertilized egg, which is also called a zygote. Okay. Do I expect you to know this term? Yes, I do. Okay. All right. So that is the breakdown of the difference between our body cells and these sex cells called gametes. Okay. So we're going to be concentrating on this side because we already looked at somatic cells and mitosis. Now we're going to be looking at our gametes and how those are formed and the importance of them. So a gamete and a gamete, so two gametes coming together, we have 23 chromosomes in each, and we get a zygote that's fertilized, which altogether has 46. Okay, this is important because if we look back here, that is what our normal body cells require for them to perform, mitosis. They need this. So in order for us to have, a cell that could produce normal body cells, we need to have a full set of 46. It's just that we need 23 from one source, 23 from another source to come together to make something that can, make more of itself. Okay? All right. So, how do we make gametes? This is what meiosis is all about, is how do we make them? Okay? We need to produce cells that have half of the amount of chromosomes that normal cells have. So instead of a cell with 46 chromosomes, we need to make a cell that has 23. Okay? We do this with the process of meiosis. Okay? In humans, most of the cells in our body are produced by the process of mitosis. Okay? Our body cells are produced by mitosis. Okay? Okay, mitosis. Whereas only gametes are produced by meiosis. Meiosis. Okay. At fertilization, we have 23 chromosomes donated by each parent to get a total of 26 chromosomes, individual chromosomes, which are chromatids, or 23 pairs of chromosomes, total chromosomes. So fertilization results in what's called a diploid zygote. A diploid cell is a 2N cell, N referring to the amount of normal chromosomes in a gamete. So each gamete, the number of chromosomes in them is 23. It's referred to as N in humans. And so we have 2N diploid. Di refers to 2. Ploid refers to set. So a diploid cell means a 2-set gamete, which results in a zygote. Okay? So a diploid cell is just a cell that has 2 sets of our chromosomes here. A gamete, which can be a sperm or an ova. Ova is another name for egg. So an egg, like a female egg, is another name. That's what ova is. Okay. Each of these contain 22 autosomal chromosomes and 1 sex chromosome for a total of 23. That's where your 23 comes from, is that 22 plus 1. And autosomal refers to like normal like body cells, like body chromosomes, and 1 sex chromosome. Okay. Each of these gametes are referred to as haploid, which is just meaning like, so we had diploid up here, haploid, the hap part refers to a single or 1, so it's a single set cell where it has 1 set of 23. N is different in different species of animals. It can be more if it's a more complex animal. It can be less if it is a less complex animal. Okay? Moving on. Like I talked about, ploidy or ploid just refers to the number of sets of homologous chromosomes. Homologous, homo, referring to same. Okay, homologous or same kind. So it just refers to the chromosomes in the cell. Okay, it refers to a set. Haploid is 1 copy, like I talked about, a single copy. N is the amount of gametes in that like set. Diploid, like we talked about, is a 2 copy like cell, also referred to a 2N cell. These are gametes. These are your somatic cells, or your body cells are diploid. Your gametes are haploids. Diploid organisms receive one of each type of chromosome from female parent, which is also known as the maternal chromosome, and one of each type of chromosome from the male parent, also called the paternal chromosome. So your mom on your mom's side is the maternal side, your dad's side is the paternal side. To remember that, matriarch refers to a female in charge. A patriarch refers to a male in charge, or like a male figure. Okay, so maternal, think of maternity clothes. Maternity clothes are only for, for women, for moms. Okay, think of that. Whereas paternal, look at papa, being your dad. Instead of maternity for this one, you could think of mama. Mama. Okay, that's how you can tell maternal and paternal apart, is by those two. And then finally, homologous chromosomes down here at the bottom. This is where we have two chromosomes composing a pair that both carry genes controlling the same inherited characteristics. This means, okay, so I'm going to explain this really quick here. Let's get rid of some of this writing here so we can concentrate. Homologous chromosomes refers to this. So that's when, we know that we have this X looking thing is our chromosome, right? Homologous chromosomes, it refers to this when we have two chromosomes together that are the same. Okay? So say this is a chromosome that codes for like eye color or something. Okay? Then the homologous chromosome to that is the one that has the exact same genes as this one does. And so homologous means they're the same chromosome, only twice. They're paired up. Okay? All right. So I just wanted to explain that vocabulary for you. Onward, here's what your 24 chromosomes look like. Each of these is a homologous pair of chromosomes. You'll notice we have 23 pairs, or 46 individual chromosomes in these X's. There's 46 individual ones and 23 pairs. One comes from mom, one comes from dad. The first 22 here are your autosomal chromosomes that will determine everything from what your body height is, your weight, your eye color, hair length, hair color, everything about what your body is in these 22 pairs of chromosomes. And like I said, one comes from mom and one comes from dad, for the paternal side. Okay? And then this very last pair over here, these are your sex chromosomes. Again, one from mom and one from dad. Okay. These things determine the, how do I want to say, what's the word for it? This determines the sex of your child or yourself. Okay? This doesn't determine your gender, because we talk about gender being different and fluid. This is determining your sex, meaning the sexual reproductive organs that you receive as a child, as a person. Okay? Humans, this is a set of human chromosomes, and the way you know that is because we have two sets here of chromosomes. Let's see. Yep. Notice you have a pair of each chromosomes, one from mom and one is dad. Diploid means you have a pair of each chromosome. Strawberries, on the other hand, are octoploidy, meaning they have 8 copies of each chromosome. They have 8 ones, 8 twos, 8 threes, etc., etc., or however many like autosomal chromosomes they have. They have 8 pairs of them. Or 8 copies of them. It's pretty crazy. And it does that because it wants to make sure that it has enough genes to make the plant and the fruit that it bears. Okay. So that's what chromosomes look like and how you discern each of the pieces and what like homologous chromosomes are and what are your sex chromosomes look like, etc., etc. Okay. Moving on. So here's a look at the difference between mitosis and meiosis again. We have mitosis, your cells are 2N, they're diploid, they're clones of each other. They have the same genetic information from the parent cell to the daughter cell. And it's just like saying, give me another one just like that, the other. Okay? Whereas meiosis, we have a haploid cell, which is just one set of chromosomes. We have daughter cells that are different from the parent cell and from each other. So whatever is made are different from what it came from, and whatever ones come in the future are different from that. Okay? Each of those daughter cells have half the number of chromosomes as a normal somatic cell, which makes sense. It's 1N, whereas that 1N is half 2N. And the whole thing about meiosis is shuffling up the genes or mixing it up. Give me something different is what meiosis is all about. Okay? So, I'm going to tell you right now that mitosis and meiosis look like a very similar process. The only difference is that meiosis is longer. Can't write. Meiosis is a bit longer. Sorry about that, folks. I had a quick visitor. Meiosis is longer in that it has more phases. It's supposed to be a few phases. It has more phases and it has more steps than mitosis does. But they look very, very similar to each other. But I'll show you the main differences. Okay. So it talks about difference in metaphase 1. when you see the word 1, or like the Roman numeral 1, you should be expecting to see a 2 as well. Okay? Which is true. The reason that mitosis is longer is that it has 2 versions of each phase from mitosis 1. Okay? So we have a metaphase 1 and we'll have a metaphase 2 later. But the difference between metaphase 1 from metaphase in mitosis has to do with homologous chromosomes compared to normal chromosomes when they line up. So for example, in mitosis, we had our chromosomes line up and then be pulled apart like this. However, in meiosis, our chromosomes will look like this when they line up. They'll line up in what's called a tetrad. It's there at the bottom. It'll go away. Tetrad, which is where we'll have our 4 sister chromatids or pair of homologous chromosomes line up like this. Okay? So here we go. So that's how our slides are going to be like laid out a little bit. So in general, meiosis is the process of cell division that produces gametes. It produces gametes and not autosomal cells. It produces gametes only. That's the whole reason why we're doing meiosis, is to create gametes and not autosomal or body cells. Okay. Our stages of meiosis 1 in for meiosis is broken up into stages for meiosis 1 and meiosis 2. So meiosis 1 is where we go from a diploid cell to a haploid cell. Okay. So we have prophase 1. This is where our chromosomes become visible, just like normal. In normal mitosis, our nuclear membrane disappears, but we have something new happening called crossing over occurring during prophase 1. Okay, and I'll talk about that here in a second. This is what it looks like. We have our centromeres moving apart like we normally do. If we were looking at mitosis, we got our chromosomes being made, like all of our DNA is winding up, that kind of stuff. Okay? We just have something new happening called crossing over. So crossing over, what is it? Crossing over is when we have genes from your maternal and paternal side crossing over. So you have your 2 ones, say you have your paternal one next to your maternal one, and they decide, hey, you want to trade genes with me? And they go, sure. And so what happens during crossing over is that part of the maternal chromosome and paternal chromosome will switch places. Okay? And the way it does that is they kind of like crisscross over and they just like pinch off where they meet. And so now the paternal will have part of the maternal chromosome and the maternal will have part of the paternal. Okay, the reason that this happens is because it increases genetic diversity, which is a good thing. Okay, so this is something that happens during prophase 1, right at the beginning of meiosis. Okay, then we have metaphase 1, which is where our homologous pairs of chromosomes line up in a tetrad at the equator of the cell. we have something called independent assortment happening. This is where the way the pairs of chromosomes line up in metaphase 1 does not affect how the other pairs line up. Okay, so they line up like this, where the homologous chromosomes line up from, making sure maternal and paternal are next to each other, one facing one set of centromeres, the other centromere. This way, we don't get two of the same on one side. Okay, that's kind of what independent assortment is, where they line up like this instead of like side to side or something like that. Okay. Independent assortment is also done to increase genetic diversity or variety. Okay, we could have the paternal sides lining up together on one side compared to the maternal side and getting them that look like this. Or we could have another possibility 2, where they line up where maybe the paternal and maternal line up on the same side, mixed up, where one color of each is on each side, and which will get us two different possibilities. Okay. And then, so this is metaphase 1. And then when we come back around for metaphase 2, we have something similar happening to get a different result. So right now we're just doing these possibilities, and then we'll end up with these later for metaphase 2. Okay, we'll come back to this in a second. Then we get anaphase 1. Each homologous chromosome moves to an opposite side of the cell. Similar to what would happen in anaphase 1, like anaphase of mitosis, the cell pulls apart, only it's with a full chromosome being pulled to each side of the cell instead of half of it. So the chromatids, each side of the chromosome, does not separate at the centromere yet. It's just the whole X, looking chromosome thing going to each opposite sides of the cell, like this. Okay? Okay. Then we have telophase 1, which is similar to in mitosis. The chromosomes gather at the poles, cytoplasm begins to divide out, and the nuclear membranes reform around that, the genetic material, the chromosomes, but they don't uncoil. Okay? They don't uncoil at this time. They stay as they are, and then cytokinesis happens, where we divide into two haploid cells. Okay? We don't have any replication of DNA at this point. We don't go to interphase. We go from cytokinesis immediately into our next phase, which is meiosis 2. So right now, all we did is pretty much let me go back here. We had our DNA become condensed, pretty much have those chromosomes line up, get separated, and now we have, in each phase, the same stuff happens. Prophase, same kind of stuff happens. You have your DNA coming out. Metaphase, your chromosomes lining up in the middle. Anaphase, things are getting pulled apart. And in telophase, the cell is tearing apart. And then cytokinesis, it actually pops apart. Okay? The only difference is that they have full chromosomes in them here instead of the chromatids, like in mitosis. Okay. From here, we go straight into meiosis 2, where we're going from having a haploid cell to another haploid cell in meiosis 2. Okay. We start again, with prophase. It's just prophase 2 at this time. Your chromatin hopefully is still in the chromosome phase because, like we said, oh, never mind, sorry, it does uncoil. My bad. Your chromosomes uncoil into chromatin as your cytokinesis happens. Sorry. In prophase 2, your chromatin becomes chromosomes again. Your nuclear membrane disappears just like it normally does, and you get centrioles forming just like they normally do. Okay? So it almost looks like two individual cells going through mitosis at this point. In metaphase 2, our chromosomes line up like they normally would in the middle of the cell. This time they do it in single file instead of as a tetrad. They'll have your centrioles move to the poles and attach spindle fibers like normal. This looks like mitosis. Two different cells going through mitosis. Okay, only it's not. This is metaphase 2. Okay. Then we have anaphase, where those two are the centromeres split, pulling your chromatids apart. So your chromosomes are getting split at this point into the two halves. So each half of the X goes to each side. Again, this looks like what happens in mitosis, where we have your X's being pulled apart, except it's in two separate cells here. Okay. Then we have telophase 2, where our chromosomes relax back into chromatin like normal. A nuclear membrane forms around them. Your spindle fibers disappear, and your cytoplasm begins to divide. Okay. And then we have cytokinesis. And each of those two that are pulled apart then become 4 haploid cells. Okay? These go on to be either sperm or eggs. Okay? And that's all there is to meiosis. Is it almost looks like it goes through mitosis twice, and instead of dividing your DNA, it just instead of replicating it, it just pulls it apart over and over again. Like it pulls it apart twice. It pulls it in from homologous into single chromosomes in each different cell, and then each of those halves of your chromosomes gets pulled in half again to make 4. Okay. So we talked about, so that's all there really is to meiosis, is it goes through the normal prophase, metaphase, anaphase, telophase phases. We go through each of those phases twice. Same with cytokinesis, we go through it twice. We only go through interphase once, and that's at the very beginning before we even begin to do the meiosis process. Okay? Okay, I'm going to talk a little bit about homologous chromosomes as well as genes and whatnot. So on your homologous chromosomes is your DNA, and the way that your DNA is wound up on each of those is the same each time for everything. This way we have things called genes, which are like the characteristics that you'd have is in the same location on each chromosome. That's why when you cross over, you don't have things messing up, because they just have the same gene in the same regions on them. Okay? So it's saying down here, sister chromatids are exact replicas, but homologous chromosomes are not. So what we're saying here is sister chromatids are the each half of the X. So this is sister 1, this is sister 2, as well on this one, this is sister 1 and this is sister 2 chromatid. But these two chromosomes are totally different. They could have a different way that these genes are written compared to these genes. However, these genes code for the same thing from each, which makes sense. Say mom had blue eyes and dad had brown eyes. Eyes color could be the same on each gene, but they're coded for different things. Okay, does that make sense? Okay. Do I want to talk about karyotyping? I might as well. So karyotyping here is, this is the what we do when we are matching up and looking at someone's DNA and looking at if they have a genetic disease or not. And the way we do that is by looking at all 23 homologous pairs of DNA that you have in your cells. Okay? So for example, we can see that you have 2 1s, 2 2s, 2 3s, so on and so forth. And you have a pair down here. Karyotyping can help with determining, like I said, certain genetic diseases, because you can tell from this guy or girl, this person here has 2 of each. Look, everything looks normal. Whereas over here, this person here, we have 2 1s, 2 2s, 2 3s, 4s, 5s, 6s, 7s, 10s, all of these. But then we run into down here where we see that we have 3 number 21s. That means that there's a problem, or that this person has a genetic disease. And if anybody knows what that is, this is where we have 321s. It's also called Down syndrome. Okay? All right. So I'm going to talk about the advantages and disadvantages of sexual and asexual reproduction now. So sexual reproduction, the main thing to it is that it provides variety. We have 3 sources where that variety can come from. Independent assortment through meiosis. So we go from having 2n in our lab to 2 to the second. So we have, 2 ways that they can be different, which is 4 different types. In humans, this means that since we have 23 separate chromosomes, we have to take 2, and each of those 23 can have those 2 possibilities. This means that we have 8 million different types or ways that an individual can form, or have like 8 million different types of person come out of independent assortment alone. Is that enough variety? Well, we said there's 3 sources of variety. This is the first one. So keep this in. So here's that diagram again where we had meiosis, in meiosis 1, this was metaphase 1. It broke those in up 2 different separate ways. And then down here in metaphase 2, when we separated, we got 4 different possibilities. Like 4 separates, 2 possibilities each. Okay. The second way we have diversity is through random fertilization. So if we look at our possibilities here with 8 million possibilities for each person, this means dad has 8 million different sperm and mom has 8 million different varieties of eggs that she could form. Ooh, we got thunder outside. This means we have 8 million times 8 million different possibilities. This means that we have 64 trillion varieties or individuals that could be formed from a single pair of human beings. Okay, is this enough variety? Nature says no and provides more variety through crossing over. Remember our crossing over? Okay. Crossing over squares the number of outcomes because crossing over can happen in each of our homologous chromosomes to produce 64 trillion squared, which is something that has 27 zeros behind it. That is 64 trillion. Okay, this is the number of possibilities of children, like differences, for every pair of like parents. So every child has a 1 in 64 trillion chance to look the way that they do. Okay? This is why many people do not show or look the same because there's so many different possibilities where children can look different from other people, from their parents, and from other people because of this amount of variety from each pair of people. That is a whole lot of variety, and this is a good thing. Okay? Why is it a good thing, though? Think about it for a second. Why is it a good thing that I look different or even have different, traits than other people? Why is it a good thing that I have, you know, certain, it's not just things on the outside, it could be things on the inside. For example, in our whole situation right now in 2020 and 21, is my immune system the same as someone else's? No, it's not, because of the variety, like the genetic differences that I have. My immune system might be stronger than someone else's, or it might be weaker. Maybe I'm more prone to getting a disease than someone else. Maybe I'm less likely to get another certain disease, though, because of my genetic variety that I have compared to someone else. This also crosses into the realm of plants as well. Our plants, like our crops, if, say, a disease came out that attacked our crops, if all of our crops had the same kind of genetic diversity or were all the same, all of our food is going to get killed off by that disease, which does not bode well for us. But if we had a variety of different plants with a lot of genetic diversity, there's a possibility that there's going to be a plant that is immune to that disease, and we'd be able to cultivate that and eat it. We wouldn't starve to death. So genetic diversity is a good thing because it makes us different from everybody else. And it also gives us advantages compared to other people or against the environment that we are trying to live in. Okay? Let's talk about twins for a second, because a lot of you go, well, some people look the same as others, which are known as twins. Okay? There are actually two types of twins, though. There's fraternal twins, which are where we have two eggs that are fertilized at the same time. And this is not as uncommon as you might think. It's especially more common in women who are older because their bodies want to give us the best chance to reproduce as possible. And so sometimes two eggs are released at the same time, and then two are fertilized at the same time. Whereas that's fraternal twins. Then there's identical twins. And this is when we have a single zygote, a single fertilized egg that splits into two or three or more different eggs, different beings. Okay, so in other words, this is fraternal twins where we have two eggs being fertilized separately, and then we have identical twins, which is down here, where one egg is fertilized, but that fertilized egg divides into two parts, and then they develop independently from each other. Okay. In the case of fraternal, they have different genes and tend to not look as similar to each other as with identical twins. They have different genes, fraternal ones do. This means that you can have a girl and a boy with fraternal twins, whereas identical twins, you can only have two girls or two boys because they share most of the same genetic material. Okay? This includes their sex chromosomes. Well, so that's about twins. That's kind of fun. And then lastly, looking over chromosomes and what genes are. So eukaryotic DNA is organized into chromosomes. Okay. Blank have specific places on chromosomes. This is genes. Genes have specific places on your DNA, on your chromosomes. And then our chromosomes carry these genes. Had another visitor. Sorry about that, folks. I was losing my train of thought. Our chromosomes carry our genes. And then finally, blank are units of heredity made of DNA. That is a gene. Okay? So genes are a unit of heredity, which we'll talk about heredity in our next unit. And those genes are on our DNA, which are made into chromosomes. Okay. So that is the end of part 2 of our notes, talking about meiosis and all the things that kind of go with it. We learned about meiosis, each of the phases of it, what they do, as along with homologous chromosomes, what our chromosomes are, what they have on them. And we learned a little bit about other things like twins and etc. Okay. If you have any other questions on this stuff, let me know. I know it seems like it's a complicated process, but it really isn't if you take a step back and you look at it for what it really is. If you're wondering if I do expect you to know your phases of mitosis, yes, of meiosis, I mean, yes, I do. I expect you to know all the phases of meiosis and what happens in each of them. What's the difference between meiosis 1 and meiosis 2, and the like. Okay, so just make sure you're looking back over this stuff and knowing that you do have differences between meiosis 1 and meiosis 2, as well as differences between like your 1 versions of all of your stages, as well as the differences as well as like what makes it the 2 versions different. Okay. All right. I think I've gone on long enough on this one. Good luck on everything. Let me know if you need any help. But otherwise, this is it for the notes for this section. All right. Okay. Talk to you all later. Bye.